end soon then. Okay, welcome back. We will just briefly go through the previous slides that we talked about Parkinson's, how Parkinson's is, I mean, what are the symptoms of Parkinson's? Then we talked about substantia nigra, how it is important for dopamine secretion and how this, element of substantia nigra is affected due to loss of proteostasis. So we saw that protein degradation is the main problem here. So we saw the different ways how which protein degradation is affected. So we talked about let's say the cell mediated autophagy, then macro autophagy, and also the ubiquitin proteasome system and the lysosomal pathway. Then today we'll be talking about mitochondrial dynamics. So you guys know mitochondria as the powerhouse of cell. This is how it looks like in a electron microscope. So it has an outer membrane and an inner membrane, and there is an intermembrane space in between. So the outer membrane has porins. It's a membrane channel which allows ions and metabolites into the mitochondria. And then there are several complexes on the outer membranes, which is present. In the inner membrane, there is a component called cardiolipin, which is also there in the heart tissues. That's why the name is cardiolipin. It is important for transport of proteins. So in the Christie, there is, electron transfer chain, and all transport proteins are present here also. And in the matrix, there is like TCA cycle, citric acid cycle, and all the genetic materials are present in the matrix. So this is how the organization of mitochondria is. And their components are in the mitochondria. So regarding mitochondrial dynamics, it's a very dynamic organelle. So it undergoes fusion and fission. So there are various proteins that are involved for it. So MFN stands for mitochondrial fusion protein. So that is what it means. MFN proteins, they are important for fusion of two mitochondria together. And similarly, there are DRP proteins. These DRP proteins, these are actually dynamin related proteins, which are essentially GTPases. So these proteins are important for mitochondrial fission. So for mitochondria to multiply in number, DRP1 is important. So these are few proteins. And then as you can see, the fusion and fission. So when they are fused, mitochondria are fused together, they form a little bit elongated structure. When they undergo fission, then there are like little small dots, spherical dots. So that is how it looks like. And for this image, they have used red fluorescent protein M cherry. So to tag the mitochondria and see how it looks like under fluorescent microscope. Most of you will be probably doing all this. So what happens, why do fusion and fission happens? So there are two views in this. So whenever there is fusion of two mitochondria, there are two mitochondria which are fused together. So let's say there is a defective mitochondria and a normal one. So due to the fusion, there will be a content mixing, and the defective part will be diluted. So that's the idea. That fusion is one of the protective mechanisms. Then there is another view also, that if there is lack of fusion, then there will be mitophagy of the defective mitochondria. So the ones which are not working properly, let's say the orange mitochondria, they will be killed. But the rest of the ones which are remaining, which remain in the cell, they are actually the good mitochondria. So again, there are two views behind this. Which which works out, that is probably something that you will be studying later. So it has been seen when DRP1 mutation was done, or if there is mutation in DRP1, what would happen?
Then
Anybody?
mitochondrial fission would not take place.
Fission won't happen, and that has caused neonatal lethality in newborn girl within a month. So it is very evident how important uh, you know, mitochondrial fission, these kind of proteins are. We'll discuss about it in detail. So here we are talking that about various mitochondrial proteins, they are dysregulated with age. So that causes a difference in mitochondrial network architecture in older organisms. So if you see aged mice, they have lower level of DRP1. So with age, DRP1 levels they increase, decrease, and aged human epithelial cells, endothelial cells, they also have a down regulation of DRP1 and FIS1. These proteins are again important for fission. So they have elongated mitochondria. So that is what is observed. If you enhance the expression of DRP1 in Drosophila, then it has been observed that the lifespan of Drosophila increases. uh, s- so that's some observation. So some uh, changes in mitochondrial, I mean the proteins which are involved in mitochondrial dynamics, if we have some mutations there, then it leads to some neurodegenerative diseases like OPA1 mutation. It leads to dominant optic atrophy. And then MFN1, as you know, MFN is used for fusion. That causes neuropathy of long peripheral nerves. So it's very important that these proteins, they remain intact in a cell, otherwise it leads to a lot of diseases. So then we will talk a little bit about the mobility of mitochondria. So you must be knowing about anterograde movement, retrograde movement. You know about it? Anterograde
Anterograde
movement
is
is
towards
away from the cell nucleus, and retrograde is towards the cell nucleus. So both of these movements are exhibited by the mitochondria. Now, the question is how are mitochondria degraded. So there is a specific process called mitophagy for this. Mitophagy is a selective autophagy for mitochondria. So the receptors are like ATG32 involved here, also NIX, FUNC1, BNIP3. These receptors are important. If these receptors are present, that means mitochondria will be degraded. So in the autophagosomes. So
Sir,
essentially,
please repeat this part. Sir, please repeat this part.
if we have, so ATG8, as you can see that ATG8 forms the autophagosomes membrane, right? Now, if you have the receptors like NIX, we'll talk about NIX in detail, or ATG32, these kind of receptors, if they are present, they will, I mean, they will make the mitochondria go for mitophagy. So the mitochondria will die, basically, essentially. It needs to die at a point. So this happens due to mitophagy. We'll talk about it again. Why is this important? So as you can see in this figure, so there's a reticulate phenotype of mitochondria in a control situation. But when there is starvation, it induces mitophagy. So that's why there is a diffused fluorescence. So the effect, the activity it reduces essentially. So this is what we see. So what is the role of mitophagy? Uh, the non-damaged mitochondria could be eliminated, or the damaged mitochondria could be eliminated due to mitophagy. So we'll talk about it. Like if we have, as I told you previously, that erythrocytes, when they mature, they lose their nucleus, they lose their other organelles, like mitochondria is also lost by the RBCs. So on that context, if there is mice which does not have nix, uh, the one of the receptors, then we see that these RBCs, they develop with mitochondria, and they have a very low efficiency. These mice they are severely anemic. So that's the problem. So this is what happens. Now, even in C. elegans, there will be some studies which was done to, you know, see whether the paternal mitochondria, they died. That all happened due to mitophagy. So there are some like receptors involved there as well. So the paternal mitochondria or the mitochondria which is present in the RBCs, these are not damaged mitochondria, yet they have to face the fate of autophagy, because we do not require them in the system. That's not how life has evolved. So in that context, when maternal mitochondria, they were labeled, you see the maternal mitochondria, they increase in number. Can you see that?
Yes, sir.
Similarly, when paternal mitochondria were labeled, in the very initial stages, when there was a paternal mitochondria, when they were labeled, then we saw that it was basically lost. So there's no green fluorescence in the on the right hand panels. In 36 hours, you see some green dots, but later you don't see anything. So this tells us how tightly regulated this process is, how important this process is for maintenance of mitochondria and the entire dynamics and everything. So we'll talk about a signaling cascade, which is a pink one Parkin signaling cascade, which is important for degradation of mitochondria. So if you remember, we talked about pink one and Parkin, both of these things in Parkinson's disease.
Yes, sir.
Okay, so we'll see how it is important. Pink one is a serine threonine kinase, and Parkin is a E3 ubiquitin ligase. So pink one in a healthy mitochondria, it is present in the cytosol. It is brought inside the intermembrane space, and that's where it sits. And then it is processed, and it goes out, and it is then degraded. That's how a normal process looks like. uh, s- and this delta psi m, this tells you the mitochondrial potential of the or I mean of the organelle basically. Now in a damaged mitochondria, this psi m, this shows the polarity of the mitochondria. It is lowered due to which pink one is no longer it's no longer brought in to the intermembrane space, and it only remains near to the cytosol. Now, this pink one, it phosphorylates itself because it is present in the outer membrane. And because of this phosphorylation, it activates Parkin, which is an ubiquitin ligase. Now, this Parkin, which is a ubiquitin ligase, now which is present in the cytosol, which is now also active, starts ubiquitinating the proteins which are present at the outer membrane of the mitochondria. And since some of you have already studied that if there is ubiquitination at specific location, then what happens? What is the fate of that protein?
The protein degrades by the
Exactly,
ubiquitin proteasome
exactly,
system.
exactly. So you through UPS it degrades. So that is exactly what happens. Few very important proteins like which we call as mitofusins, important for mitochondrial fusion and fissions. These proteins, which are also present on the outer membrane of the mitochondria, these kind of proteins, they degrade because of this process. So this is how the entire schematics look like. So when pink one is present, it you know the parkin is not working properly. But if pink one is phosphorylated, what happens? It activates the parkin. Phosphoparkin act is active, and it goes on ubiquitinating. These boxes are ubiquitin moieties. It goes on ubiquitinating the membrane proteins which are present in the mitochondria, which leads to again degradation, as you people, some of you already pointed out.
Sir,
So
what is this phosphoparkin? Please go to the previous slide. Sir, here auto-inhibited parkin means, I mean, like if the parkin is not
not
function.
phosphorylated. Pink
Achha,
one is a kinase. Kinase phosphorylates, right? So if
yes, sir.
kinase is not available for phosphorylation, what would happen? It cannot phosphorylate parkin. But
Yes,
since
sir.
the mitochondria is damaged, it's present in the cytosol, it cannot be taken inside. So this pink one, it phosphorylates the parkin, ubiquitin ligase. And since it is phosphorylated, it starts phosphorylating whatever is present on the surface of the mitochondria. And the mitochondria, in the end, it suffers the fate of autophagy. So that's the idea here. So these are so well regulated processes. It happens on a mitochondrial level in each cell, and we are not even aware of it. So that's really a great thing. So we have already seen that lysine residues, K residues, they are usually ubiquitinated, and each uh, lysine residue which gets ubiquitinated, like polyubiquitination of K48, lysine 48 residue of lysine, it leads to ubiquitin proteasome degradation. Whereas if there is ubiquitination of the 63rd residue of lysine, then it leads to signaling. And there are some atypical ubiquitination also, which leads to fast track degradation. Ubiquitination is also important out of the mitochondrial context. Let's say if there are some lysines, if they are ubiquitinated, then we see that it leads to DNA repair and some things like that also. It's not that ubiquitination always leads to degradation, but definitely degradation has been very well studied in this context. So this is exactly what it explains what is happening. So in a depolarized mitochondria or a damaged one, the import of proteins is abolished. Import of proteins like pink one is abolished. So as a consequence, the pink one, it accumulates on the outer membrane of the mitochondria, and its kinase domain is facing the cytosol towards the cytoplasm. So this Parkin recruitment, it activates the mitophagy process. So when it is translocated to the damaged mitochondria, Parkin ubiquitinates the outer mitochondrial membrane proteins, and this causes Parkin-mediated polyubiquitination of mitofusins, and it leads to proteasome-dependent degradation. So we talked about it.
Sir, can you go to the previous slide once? Wait, sir, 1 minute.
Any question?
Sir, here it is written that its subsequent stabilization on the outer mitochondrial membrane results in parkin recruitment and mitophagy activation.
Hmm.
Uh, Stabilization of this pink one.
Yeah, pink ones, it stabilizes in the outer mitochondrial membrane. Its active site is towards the cytosol. So since it's stabilized, now it recruits Parkin, it phosphorylates Parkin. So what you have
Sir,
seen here,
here
the same thing is explained.
that means, sir, pink one means what is the basic function of pink one? It is a kinase enzyme, right? So it phosphorylates the parkin protein. I mean, its function is to phosphorylate the parkin protein.
No, in a healthy mitochondria, its function is to phosphorylate, but it happens in the intermembrane space.
Okay.
So here a lot of things happen, even ATP production, a lot of things basically happen. So their phosphorylation is important. For what reason? That's not been, I mean, discussed in detail, but it is important in normal mitochondria. But since in the damaged mitochondria it cannot get in, it starts, active. I mean, phosphorylating the proteins like parkin, and this parkin starts ubiquitinating, and it leads
And sir,
to
what is this? What is the function of Parkin protein then?
parkin is a ubiquitin ligase.
Acha, acha. Okay. Oh, right, right. E3 ubiquitin ligase. Yes. So that means, and pink one cannot set it into the inner mitochondrial membrane because of the fact that the del psi m, that is the chemical potential of the mitochondrial, the perimitochondrial space gets decreased, right?
Right, right. You are right. So what does the mitochondria do? What's the main function of a mitochondria?
So ATP generation, energy generation,
How does it do it?
with the help of oxidative phosphorylation in
That
the
is I
electron
that I
transport
know,
chain
that I know.
occurring in the
What is
perimitochondrial
the
space. Acha.
force that a mitochondria uses? What is the energy that is uses for ATP generation?
Hydrogen proton gradient, proton motive force.
Proton motive force. So if a mitochondria is not functioning properly, this gradient will be
Okay,
lost, right?
understood. 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 Okay, yes. Less amount. The proton gradient gets decreased actually. Okay, understood. Understood. Del psi m decreases. That means the proton gradient is also low, very low.
So it essentially the polarity it of the
Okay.
mitochondria it no longer remains. So as I said, polyubiquitination of mitofusins it leads to proteasome dependent degradation. And so what happens if mitofusins they are degraded? So mitochondrial fusion no longer happens. So the fusion of the mitochondria it is prevented in this process. So again, mitochondrial degradation it could happen in two ways. One is the Parkin dependent independent path should be Parkin dependent pathway. One is Parkin dependent, another one is Parkin independent. So in the Parkin dependent pathway, there is pink one, symphyllin 1, which is important. Even in cancer cells, it has been observed where Parkin is frequently downregulated. Uh, uh, there is another protein that polyubiquitinates the mitochondria and it induces mitophagy. And then there is receptor mediated mitophagy where pink one phosphorylates ubiquitin, and this phosphorylation event it triggers the recruitment of autophagy receptors, and then the mitochondria is degraded. So and that's all we have. So what we observe with aging is that there is a dysfunctional mitochondria. So usually how we see fusions and fissions of mitochondria occurring, that entire dynamics is changed in case of aged patients, and it leads to a lot of disorders like metabolic disorders and neurodegenerative disorders and cardiac diseases as well. So mitochondria it plays a very very important role in maintaining the cellular energetics, in maintaining the yeah, I mean the cell, the energy of the cell and everything else. So that's how it works. So in few studies, if you remember earlier also, it was observed that when mitochondria is not functioning properly, then the number of mitochondria it increased. So it led to a lot of mitochondrial fission, but that in an effect, it did not work out on the long run. So things like that happen with age. So any questions so far?
So then that means with increasing age, mitochondrial fission rate increases.